First, I'd like to thank Flavie and Doug and Sarah for the opportunity to get you up to date on gene therapy. I think we have made significant progress, and many of the questions that were raised here in this session could be readdressed at gene therapy. I mean, if we replace the gene, how much can we restore, at what stage we can restore, and so forth. So. What I want to talk about in this session is the choice of virus for gene delivery, which Doug alluded to. The methods of gene delivery. This is very important. The challenges posed by the large size of the Dysferlin gene and choices of potential genes to deliver. And I really will give you a perspective of what we have explored clinically over the last decade. First of all, it's very important. I think one of the most confusing things is this term adenovirus versus, I guess that's not showing, so I need help with that. There it is. I think that'll do it. Yeah, yeah. So adenovirus and adeno-associated virus. These are two distinct viruses, and they have some collaboration at times, but for gene delivery, they are very different. There was a major problem at one time when I first got into this field in 1999. We did our first gene delivery study. That adenovirus was still popular, and prior to that we had planned to take the very large Duchenne gene and put it in adenovirus, but because of a major complication in a patient, that was really abandoned pretty much for gene delivery. Adenovirus is often used for cancer and some diseases, but not for single gene defects like dysferlin. Lentivirus is an interesting one. We use it a lot in the lab. It has an advantage of a very large capacity for holding genes. The problem is it integrates into the genome, and by integrating into the genome, it can create problems and disrupt other genes, and it has actually done that clinically. So adenovirus really is our target, and it's the choice of virus for gene replacement because it causes no human disease. This is very important. All of you have been exposed to adeno-associated virus, and we call it AAV, and if we check your blood, we would find exactly what type of, or if you've been exposed to adeno-associated virus. It does have a relatively reduced immune response, but it's not clean, and we have seen this especially in some of our recent studies. It has very little toxicity in itself. If we infect a person with AAV, we see virtually no complications. It's delivered to the nucleus, and in the nucleus, this is a major advantage. It has no integration into the normal genes, into your other genes or into your genome. Its biggest problem that we've had to face is that it has a small insert capacity. And I use an example, it has a capacity less than 5 kb, and for example, the dysferlin gene needs 6.2 kb, and how we deliver that has been a unique process that I'll talk about. But basically, we get asked a lot of questions about how we do this, and the way we do it is conceptually very simple. We take out the viral genes, and in AAV, there are only 2 genes, and so it's a relatively simple process. And in place of that, we put in the gene of interest. I show here dysferlin, but we've done multiple clinical trials. We're not quite yet ready for a clinical trial in dysferlin, but we're definitely targeted that way. Now, talk about the methods of delivery. Doug alluded to this, and we've used now multiple methods of delivery that will pertain to dysferlin gene therapy. we can deliver the gene. The simplest way is to deliver it directly to the muscle. And often the FDA requires us to do this. It may require this in dysferlin because dysferlin, the gene delivery in dysferlin is such a unique process. It's never been done before clinically, and the FDA mandates safety as #1 priority. And so in first clinical trials, we often have to do intramuscular gene delivery, and I'll show you how we've done this in the past. The first gene therapy study that was ever successful was in another form of limb-girdle dystrophy, 2D. And I think most of you are familiar with this. Here's 2D. It's alpha-sarcoglycan deficiency. Here's dysferlin right above it. But these are relatively two of the more common types of limb-girdle dystrophy. Singularly, dysferlin is more common than alpha-sarcoglycan. Collectively, there are a group of sarcoglycans that may compete with the incidence of dysferlin. We're often asked about, where do you get the genes? How do you make these things? I mean, this is a common question we get in the clinic when we're seeing patients. And we build what we call a cassette. This is a cassette of DNA. And we can put this into DNA production system and generate exactly what we want to. We know the whole sequence of the human genome, so it's not really a problem. And in this case, this is the alpha-sarcoglycan gene, and here you can see we can generate the alpha-sarcoglycan gene. That's, when we say that, we're really putting into the cassette, as we call it, the actual coding sequence. This is beyond the exons now. This is into the coding sequence of the gene. And we've had a lot of experience with exon skipping, so I'll be glad to address any questions about that, but this is quite different. But notice also that the size of the sarcoglycan gene, it's 1.16 or 1,000 base pair. Remember that we can insert into the AAV about 5 kb or 5,000 base pairs. So we're well within our range of inserting this cDNA. It has to have certain other things. Every gene in your body has to have a promoter, and no gene works without a particular point that allows for the gene to express itself. And in this case, we have an MCK. CK has come up multiple times in this discussion, and this is a muscle-specific CK promoter, and we like to use a muscle-specific promoter because if the gene gets elsewhere, and it will get elsewhere, it won't express. And if it doesn't express, there's no toxicity with it. And it has what we call a poly A tail. Every gene has a poly A tail. And so the whole thing here is not 2 kb, and so it's perfect for fitting into AAV. And in this case, I'm going to show you a study that we've done with AAV1 and an MCK promoter and the alpha-sarcoglycan gene. This was actually the first gene therapy for muscle disease that has ever shown efficacy, but it was in a limited fashion. And it was in a limited fashion in that we injected it directly into the muscle. And this is a muscle that we like to use. It's called the extensor digitorum brevis. It's a foot muscle. It has basically no important function. It's a residual muscle in evolution, and we can remove this muscle. It's a small toe extensor muscle, but it doesn't cause any problems for the patient. And here I am injecting the gene into the EDB muscle, and side by side working with an interventional radiologist. And the interventional radiologist can show me exactly where we have the needle inserted. This is inserted into the EDB so we can see that. And what we do is we inject the gene parallel to the muscle and then perpendicular to the muscle, so we have a heavy concentration of the gene right in the center of the muscle, And so we can take out the muscle then at various times. We've taken it out as early as 6 weeks, as late as 6 months. And here you'll see the 6-month result in 6 LGMD patients, 2D patients. We have biopsies we did at 3 and 6 months, and we have perfect, beautiful gene expression in about 70% of the muscle fibers. And in contrast, what we do in cases like this is we'll inject the gene on one side, inject a control or saline on the other side. It's blinded. I don't know which one. I'm handed a syringe and say, inject this one in side A, inject this one in side B, and at these time intervals, we take out the EDB, and we can see in the EDB differences, we hope. If there's no difference, obviously there's a problem. The gene didn't get delivered, and we've seen that before when we have neutralizing antibody. If a patient has high neutralizing antibody to AAV, the gene won't get in. It'll be blocked immediately. So that can be a problem. So this was a successful study, and it was very gratifying. And based on that, we've gone on now to do the first intravascular study of alpha-sarcoglycan. And how do we do that? A method that we use is called isolated limb perfusion. And we like to use isolated limb perfusion because it concentrates the gene in a localized extremity. So we can put the gene in, get very high concentrations of the virus in one extremity, and it can go to all the muscles of that extremity. And this was really a major breakthrough. We've only done it actually within the last, in the last 2 months. We started 2 months ago. We'll deliver the virus to all the muscles of the extremity, and we're currently using this as an approach to 2D. Whether we use this as an approach to 2B or whether we do systemic delivery would be a matter of choice as we move forward and see how much we can deliver in systemic circulation. But this is an example of how we do it with isolated limb perfusion. And don't forget, we can do as many limbs as we want. We can isolate any limb we want or multiple limbs, even at one sitting. So it could have a profound impact. And in this case, we isolate the femoral artery, and we inject it in, put a catheter into the femoral artery that goes to most of the muscles of the lower limb. And we can actually thread the catheter down, which we have done in non-human primates, and deliver it to specifically. For example, in Miyoshi, we could deliver it to the gastroc if we needed to, or the posterior muscles of the calf. And we take this, what we call a Ty-Shak balloon catheter, and this is actually from a patient that we delivered the gene to. Here's the balloon catheter blocking the limb from any circulation that comes in, and we actually block the exit. The vein is also blocked at the same time. And so we really do have a very good system to isolate the limb. And that makes the whole process safer. But another aspect of that, it makes the process more accessible because we have to make virus to get to the entire, theoretically, to every single muscle of the body. And in many Dysferlin patients, for example, they have very good upper limb strength and very weak lower limbs. So it may be applicable, but again, these are decisions we'll make along the way. And here's the system that we used. Now, it requires a major team to do this. I showed you with an EDB injection working very closely with an interventional radiologist. But here we're working with the interventional cardiologist. This cardiologist here does all the cardiac catheterizations for Nationwide Children's Hospital, an internationally acclaimed cardiologist. And he has a team that works with him. The patient's over here asleep, and he's got the catheter in there. And my role, relatively unimportant here, is to take the gene out of the package that we get from the pharmacist and pass it off to their team, and they inject it. And in this case, we had absolutely no complications, which was really a major breakthrough for gene delivery here. And here you can see this is the catheter that's in the femoral artery, and here's the degree of gene expression that we get from a muscle biopsy upon doing this. So we get very good gene delivery, very safe, and here's the patient's extremity post-gene delivery. There's absolutely no indication, no swelling, no discoloration of the limb, and most remarkably, when the patient woke up, they didn't have a stitch of pain. There wasn't any discomfort in the limb. So we were very gratified to do this, and this is really, again, like I said, the first time this has ever been done. This is ILP. It's safe and effective. Isolated limb perfusion is what we call it. Now, the next approach to deliver the virus is systemic, and we've done that too. We're in the middle of a clinical trial, and we've done this 8 times now. And this is in babies now with spinal muscular atrophy. And this is one that we did about 10 days ago. And I took a picture here knowing that I would be here so you'd have some appreciation. And this is actually the baby getting the gene at the time we're doing it. This baby's awake. Putting it in intravenously doesn't bother the patient at all. We usually put in 2 catheters, one in one limb and one in the other, and the reason we do this is we deliver the virus over about an hour, and we try to deliver it. If we have any problem in delivery, that means that what we'll do is we'll switch to the other limb, and we don't like to be taking the time and calling our IV delivery team in Children's Hospital up to put in the catheter for us. So everything is prepared. We get the virus from the pharmacy, and in this case, I am injecting this with intravenous delivery. Now, I think Doug mentioned about immune responses, and I want to show you what our experience has been with immune responses. Now, we know that the patient's gene must be a perfect match to prevent rejection. And we had this in Duchenne dystrophy in our first clinical trial. If the patient, and also if the patient has been previously exposed to AAV and at high levels, the gene could be rejected. And we actually have set the standard now. For better or worse, the FDA now makes us use a certain amount of neutralizing antibody that we can identify in the blood, and it's very low, so we don't have rejection from the virus. But what we found very unique, and we published in the New England Journal, was the result of our first DMD gene therapy trial. And here we had a surprising immune reaction. There was never predicted to be an immune reaction. I won't go into the details tremendously in the sense that the dystrophin gene was always said to be protected because there were these fibers that would express spontaneously some dystrophin. So people said, don't worry about it. You can put in the dystrophin gene and it will be safe. It won't have an immune response. Well, hello, that wasn't true. And this is an example that I'm showing you here. This is a patient who this patient has a deletion down here. This is a 3 to 18 exon deletion. The numbers are not so important. It's more important where the gene expressed. This is our cassette. I have it divided up here in showing where it expresses specifically in the gene. And the reason for this DMD DNA or cDNA or gene transfer that we're using is that the entire gene, Duchenne gene, is 11 kb. Remember, we said 6.2 for dysferlin, although that doesn't count the promoter size, so it really gets up to about 7 that we've got to consider for the dysferlin gene. But the Duchenne gene is the largest gene in the human body. And so we can't fit it into AAV. We have to make something that fits into AAV. And this is the cassette that fits into AAV. The important thing is when we express this into a patient's gene with a deletion, you can see it expresses right into this area of deletion. And so that means that all of these protein fragments here that are expressed now are foreign proteins to the patient. So guess what happens when you get injected with a foreign protein? Your body's smart enough to develop an immune response. Your body doesn't want to see this foreign protein. And so we have a marked inflammatory response that we can measure with something called an LE-spot assay or an interferon gamma T cell assay. The name of it's not important, but we can measure this, and we do in all of our gene therapy trials. So it was a great lesson to learn, and that's what we learned in the Duchenne study. Now, the next thing is to consider challenges posed by the large size of the gene, and this is where I want to bring it home to you in terms of dysferlin. And I could talk about Duchenne, but it's more important, I think, for you to see what we've done in dysferlin. Now, what are the ways of accommodating this? Genes of transfer must be capable of fitting into AAV. And what are our choices to make that happen? One choice is to reduce the size of the gene at the risk of not correcting the defect. And this is the problem in Duchenne dystrophy. Quite honestly, we use micro or mini dystrophin genes, and when we bring them to clinical trial, which we're trying to do right now, we don't really know how much it's going to correct. We think it's going to correct because it looks good in the mouse, but does it really correct when we put it in the patient? That's to be determined. The other possibility is packaging the gene in separate pieces that are more reliable for correcting the defect. And why are they more reliable? Because we developed a system, and this was done in our lab. Louise Rodino-Claypack led the effort, and she's doing a great job. And I'll just show you. Here's the dysferlin gene that we're hoping to correct. Just bring it back a little bit home here. The dysferlin gene is found. This is the position of the dysferlin gene on chromosome 2, and that was discovered before even the gene was known. And then what we've got is what we do. We have to have the choice of virus again for gene replacement. I wanted to emphasize this just to make a couple points that I probably forgot along the way. AAV is very efficient for gene transfer. That's unequivocal. But we have to get something in the AAV that's going to work. And it's episomal. Episomal means it does not disturb other genes. It doesn't integrate into your genome. It stays in your nucleus. And to me, this is, as an old timer in this field, it's one of the most magical things you could ever imagine. It sits in the nucleus and expresses there for an indefinite period of time. And we have genes in the lab that have expressed for over 2 years. Do we have anything longer than 2 years? There's been some experiments, not in our lab, but in others, that shows that the gene can express for as long as 15 years. So we're fairly confident that that can happen. So we can have long-term persistence with a single injection. And that is one of the major advantages in doing this kind of therapy versus exon skipping, versus stop codon read-through, or others where the material has to be given weekly. We're doing a big exon skipping trial in Duchenne, which has had some success, no doubt, but we have to give it every week to these boys, and without any stopping of the delivery. And then, of course, we have the size limitation. So how do we cope with that in dysferlin? Here's the dysferlin gene that we make in the lab. It's 6.5 kb. That's greater than 4.7, as I've mentioned multiple times. We're using an MHCK7, slightly different than the MCK promoter we used in the alpha-sarcoglycan trial. It'll be the first time this one is used in clinical trial, but we're very excited about it because it has potential to express in the heart, which is a major advantage for this particular promoter. And then, of course, it has the poly A tail, and we put an intron in there which is a non-coding sequence which helps it express better. But that is over 7 kb now, and that's not going to fit in AAV. So what was done to make this happen? We stumbled on this in a sense inadvertently. We were told in the literature that AAV5, there are the multiple different serotypes of AAV, and AAV5 was supposed to package a larger genome. But when we did it, we found that it actually didn't. So we started working on something that would work better with intravascular delivery than AAV5, and we're working on one that's very close to AAV8. We could use AAV8 or AAV9 or even AAV6. All of them work well when we inject them into the circulation. And we package this in two different AAVs. One packages the 5' or the initial half of the gene, and the other one packages the 3' or the other half of the gene. And by magic, I swear this is magic, because when you put this in, these two components, and Louise worked hard on how to make this happen. And the genes have to overlap by 1,000 kb, by 1,000 base pair. I'm sorry, 1,000 base pair. And if this overlaps by 1,000 base pair, the gene will produce by homologous recombination a full-length dysferlin protein, unequivocally full length. No other fragments then are expressed. This is an absolutely incredibly perfect system. But why it works is really still a bit of a mystery, because we've tried it with other genes that don't work quite as well. And you can inject this into the circulation of the arm and find that the gene is perfectly fully expressed in the muscles of the leg. Now, if that isn't magic for an old-timer like me, nothing is, because I mean, this is incredible. And you guys, one thing, you guys are blessed by the fact that this can happen. And this is what it looks like when we express this and maintain it for a year in the mouse. This is the knockout mouse for the dysferlin gene, and we can show good expression at a month, 3 months, 6 months, 9 months, and 12 months, and that is extremely gratifying. And we've checked this as well in the monkey, and with non-human primates we can achieve similar things. Now it's tricky in the non-human primate because they're not dysferlin deficient. I won't go into the technology of showing you how we can do that, but trust me, here's in the mouse who has no dysferlin, we can show perfect gene expression, perfect homologous recombination, no toxicity, no immune response, and this is truly a blessing. Now, also, if I can make this happen. No. It's not critical, but it'd be nice just to show you. This is the membrane repair that Doug was talking about. Come on, baby. There we go. There we go. This is an incredible slide. On the left-hand side, we have a normal muscle fiber. On your right-hand side, this is what happens when you laser injury a normal muscle fiber. You get this defect in the muscle membrane that will repair itself very rapidly, but it's relatively small. And talk about calcium. Doug talked about calcium entering the cell. It'll enter the cell now through this area of injury to the membrane, and that has to be repaired very rapidly. Now, this is in the knockout dysferlin mouse, and when we do that, we have a much larger defect that doesn't repair itself, and this leads to calcium entering the muscle fiber and actually ultimately leading to the death of the muscle fiber, or at least a fragment of the muscle fiber. And this is part of the problem the membrane repair process is impaired. And when we go ahead and replace the gene, what we see is a defect that is pretty much analogous to what we see in the control, in the normal muscle fiber. So we know this works, and that is a very gratifying process. So that's the story for how we're going to replace dysferlin. It looks very promising. Where we are now is we've gone through the FDA steps, and I'll show you at the end of this talk, just in a couple of minutes, about where we are in that. Now, so replacement for 2B looks very favorable, and we anticipate moving this to clinical trial in 2016. And we know the steps in to take this to clinical trial. We've done it more than any place in the entire world. And so I think we have a very good working relationship with the Jain Foundation, and we're very grateful for their support. I want to show you one other thing that has to do with, I don't know whether Doug was alluding to follistatin or the myostatin pathway. I assume he was when he was talking about sarcopenia. And we've done this clinically now. And if you can't replace the gene specifically, you can develop a strategy that will allow you to increase muscle size and muscle strength. At least it works in the mouse and in non-human primates, and we have evidence that it works in Becker dystrophy. So I'm sure you, somewhere along the line, have learned about the myostatin pathway. If you disrupt the myostatin pathway, you produce a mouse that's very large, called mighty mouse. This is work that's been done at Johns Hopkins, incidentally, by Se-Jin Lee. Now, there's also a double muscled cow that is used in production of increased muscle for meat production, and this is what that looks like. And also, there is a human mutation that has been described in newborns that shows enlargement of the muscles. So it's true across the board. It's true for mouse, it's true for cattle, it's true for humans. So the pathway is very realistic. And what happens with myostatin pathway? It's called a negative growth regulator. And here's circulating myostatin. It forms a dimer and stays localized to the muscle so that the muscle doesn't grow to any grotesque size. And if you disrupt this pathway, and we've done it in multiple ways, but we found that follistatin is the most potent pathway, most potent peptide we can use to block the myostatin pathway. So here's the normal muscle fiber. That's what happens when you use follistatin. It blocks the pathway. So what can we do with gene therapy? What we realized is that we could put the follistatin gene into AAV, and we inject it into AAV, and the AAV then will go to the nucleus and express follistatin. When it expresses follistatin, it'll block the pathway, leading to, theoretically, when we were in the theoretical phase of this was our hypothesis, and then we took it to the next phase. We did it in mice, and I won't show you that. But what I was working with Brian Kaspar, and I said to Brian, look, I'm not taking this to clinical trial unless it works in non-human primates. So here we are in non-human primate. Here's a monkey leg, and we injected this 3 sites into the monkey leg, and we see that the muscle grows. Here's the normal control. Here's the gene leg that we put the AAV in and grew to fairly large size. And even when we put in a muscle-specific promoter, it grew in size. And here you can see a slide comparing the muscle fiber size in the gene that we injected versus a control. And here I am injecting the gene into the thigh muscle of a patient who has Becker muscular dystrophy in this case, and working side by side again with an interventional radiologist. I can't emphasize and I can't tell you how important teamwork is. Teamwork is the only way you can get things done in an efficient manner. And here we have injected 6 patients. This is the first 3. We injected 2 doses. One improved 125 meters in the 6-minute walk. Another improved 58 meters in the 6-minute walk. And one didn't respond. Well, that's clinical trials. Not everybody responds. And then we did it at a higher dose, and we found pretty much similar results. One patient improved 108 meters in the 6-minute walk distance. Another one improved 29, and one did not improve. So out of 6 patients, 4 improved. And when we mean the 6-minute walk distance, it was a steady progression that we saw over a 6-month period. So this was very gratifying. And this is potentially applicable to any muscle disease. It's applicable to dysferlin for that matter. We would rather replace the gene directly, but we have a backup plan. If that doesn't work, we have the potential to use follistatin gene therapy. And at that time, we were doing direct muscle injection. Now we've done ILP. I would prefer to do ILP if we had the opportunity. But it really depends. We're doing this in Duchenne dystrophy now, and we're injecting multiple sites. We're injecting in a Duchenne boy, we're injecting 18 sites in one leg and 18 sites in the other, and that's a total of 36 sites. The boy is asleep. When we wake him up, he has no memory of getting any injections, no pain or discomfort. So I'm hopeful that'll work in Duchenne dystrophy. We'll see, though. I just want to mention in conclusion here about the path to gene therapy. We have a great deal of experience with this and a great deal of experience with the FDA. The first thing you have to do in any gene therapy approach is establish unequivocal proof of principle. The proof of principle is usually done in the mouse. It could be done in other species, but the mouse is the common one. And this is true now for the Dysferlin gene. And then you go to the FDA with this proof of principle. It has to be solid and unequivocal. And they will help you develop a clinical plan that will include the age of the patients, the inclusion criteria, and everything you need to do for the clinical trial. They'll also help you plan a toxicology study. The mice are the usual site for toxicology studies. So we take the same virus we're going to inject into patients, and we inject them into mice. But we usually use a dose that is 10 times the dosing level we use in patients. And this will assure the FDA that you have a safe product. The next thing is to actually perform the tox study, and this is an arduous process. It's done by usually a CRO. The total tox study is not done in your institution. It's done outside your institution to show safety, and you want it done by an impartial observer. The next thing is to present the study to the Recombinant Advisory DNA Committee. This has been done for Dysferlin now, and we had no kickback, so to speak. They accepted the plan. They accepted everything that we're planning to do for Dysferlin. We're in toxicology study now. We're performing the tox study, and usually we'll present it to the Recombinant DNA Committee as we do the tox study, and they'll say that it's either approved or not approved. If it's approved, it's dependent on the results of the tox study. And then we apply for approval to our internal review board. That's in the hospital where the study is going to be done, the IRB, and most of you in some way or another are familiar with that. And then finally, the final step is submitting the IND. And I show this picture for impact because that was the picture that we used. This is a tremendous amount of work, and we have to have support to do this, and presume the Jain Foundation is up to helping us get here. I have no reason to believe otherwise, But it is a lot of work. It's a lot of work on me, my team, the regulatory core that we have in the center. And incidentally, we make our own virus for gene delivery. That's a big one. We have our own viral manufacturing facility that really cuts down on any costs. We can do it at the least amount of cost. If we go out to industry, we'll pay at least 10 times the amount of money that it would cost. So this is the team. We have support from the Jain Foundation. We've had support from Parent Project, from our Wellstone Center, and it's overall been a very gratifying process as we move forward, and I can open this up to questions. So thank you.